Just pass them along so everybody can see it.
Good afternoon, everyone. And good afternoon to everyone online. It's great to have everybody here. I just look at that real quick. I just want to see. Just double checking here that so Ken, thank you for being here. We may have Camilo join. Just want to say that one of Sarai's committee members, Camilo Trench, is hopefully joining us, but was in Jamaica for the recent storm. He's okay and his family's okay, but there's been a lot of devastation. And I just want to acknowledge the outstanding effort he was trying to go into a place where he had connectivity so that he could chime in for today's defense. I'd also like to thank the other committee members, Dr. Ken Krause, who's online, and Ali Girdal, who's the program manager for Grow USVI Mangroves, who are also in the room here today. Ken's online. It's my pleasure today to introduce Sarai Hutchinson. Sarai is a graduate of UVI's biology program. She majored in biology with a minor in environmental science. And then took a year, she was over in St. Croix working for UVI Extension on bugs and agriculture, and never far away from plants, I'll just note. And then was also worked with the Nature Conservancy and their coral land-based nursery for a year before she entered our Master's of Marine and Environmental Science program in the fall of 2023. While in the program, Sarai has done many things, but she is an NSF Securing STEM Island Pathways Graduate Fellow, which is a special fellowship that we have for UVI undergraduates who want to pursue their master's here at UVI. And she's done, I think, just about everything in the lab that there is to do. She's volunteered for, from the Territorial Mangrove Monitoring Program to our Mangroves in the Classroom program to other education and outreach within the community. So it's just been a delight to have her. And today she's going to be talking about her experiment, which I'm super excited about, but looking at novel uses for waste substrates like recycled crushed glass, but also nuisance species like Sargassum. So I know she's excited to share what she's learned over the past few years, and so I'll hand it over to her. Just one note for committee members who, sorry, this is my second one of these today. For committee members who are here, if you could please hold your questions. Sarai will present for about 35 to 45 minutes. There'll be time for public questions. At that point, I'll just ask that her committee hold their questions. We'll go into a closed door session around the hour mark where we'll get to explore all of those questions in greater depth with Sarai then. All right, without further ado. What?
Hi, and a pleasant afternoon to all. Like Kristen just mentioned, my name is Sarai Hutchinson, and I am a part of the 2023 cohort of students in the Marine and Environmental Science program. And today, I'll be presenting on how we can use sargassum and recycled crushed glass in a mangrove setting to grow red mangroves and white mangroves. So before we get started, we need to learn what mangroves are. Well, mangroves are trees that are in intertidal zones, and they compose of trees and shrubs along the coastline in much of the tropical, subtropical, and warm temperate world. They tend to live along the shoreline, and they have a typical zonation of them going from low saline areas with the more terrestrially based ones, like white mangroves, to black mangroves in the center, and then red mangroves along the coast. However, in the United States Virgin Islands, these mangroves do not tend to follow this zonation pattern. They just sort of live wherever they would like, and so we would have both red and white mangroves living together in certain areas. Mangroves provide many benefits, but however, there are many threats that they have. So mangroves provide many benefits to both the environment as well as humans, but what do we do for mangroves, right? Some of the biggest threats that I'll be focusing on today are hurricanes as well as sargassum inundations. In 2017, September 2017, the USVI was hit with 2 Category 5 hurricanes back to back that absolutely devastated the territory. From both humans in the regular life happenings to our mangroves were devastated, especially so on the image on the left. On the left, there was massive destruction at Coral Bay, and there was limited recovery, natural regeneration after these hurricanes. And so this prompted the need to have active restoration. This is where we have land-based nurseries come into play. They help to supplement this regeneration by growing them in a controlled environment. And however, doing research is fun, but it's very expensive sometimes. And a big expense is importing the substrates that are required to grow these mangroves in. Another thing I have to mention is that in St. Thomas and across the territory, the landfills are at capacity. So with things like waste and nuisance species, and so we need to then find a way to repurpose these and to give them new life. And one potential way could be using them in restoration. Across the territory, the Caribbean, and the world at large, we have mangroves along the coastlines. And we also have very similar issues in small islands as well, which is issues with waste and sustainability. And so finding ways to repurpose these things is important. One such issue is the Sargassum inundations. And starting in 2011, it has increased in frequency and abundance across the Caribbean. And these massive influxes are sustained by increased nutrients from things like the Sahara dust, as well as shifting ocean circulation patterns. When there's too much sargassum, the environment as well as the aesthetics of the beaches decline. And so it's important that we find ways to remove them that lead to too much negative environmental issues. With these inundations, we also have effects on our mangroves, because mangroves are a forefront species that protect our coastlines. And so when they begin to decompose, as we've all realized, when the sargassum decomposes, it smells really bad. That's the hydrogen sulfide. So if we can find ways to get all that out of there and make it have a benefit, that would be pretty amazing. Some research that was done in repurposing this waste is by making a compost in studies by Trent et al. 2022 and Williams and Big in 2010. And they found that it was possible to make a compost that grew red mangroves in, as well as making things like fertilizers. However, they found that because sargassum is a pelagic species, so it floats on water, it absorbs lots of salinity salt, as well as harmful trace elements like arsenic and lead. But as much as that's bad, mangroves are amazing. They're able to deal with some of these issues, especially like salt stress, because they are able to exclude and prevent the absorption of this salt. Another opportunity for repurposing of this waste is by using it to make crushed glass. In the territory, we are very tourism centered, and a byproduct of tourism is waste glass bottles. And in the territory, we have many different organizations working together to help to repurpose this glass bottles into something beneficial, like crushed glass. This crushed glass is also possible to be used in nursery propagation techniques. 2 studies that use this crushed glass were on the left, the one by Frunberg et al. 2025, where they grew black mangroves in mixtures of glass as well as river dread, which is their natural, normal substrate for restoration. And they found that yes, you can grow black mangroves in this glass substrate, but that it had negative impacts on the fine root biomass, so the very fine roots that are important for absorbing nutrients. On the right, we have the Allison Holderwood study, 2023, where she grew red mangroves in soil versus glass. And she found that yes, they can grow in this glass substrate, but they did not grow as tall or as much as the ones in soil. And so it begs the question that it may have needed some nutritional additives into this glass to make it better. This is where my study comes in. I aimed to do a study where I used the sargassum as a substitute for fertilizer in the glass to hopefully make it better. This is where I'm trying to fill that gap. Tiny break for fun sargassum pun. Sargassum? No? Okay. So for our two main species we had in the experiments, I focused on the red mangroves because one, it is the most grown mangrove for mangrove restoration, as well as I'm building upon the Holywood et al. 2023 paper where she grew them in red mangroves in the crushed glass and soil mixtures, as well as second species being my white mangroves or Laguncularia racemosa. And this is because they're the second most abundant species in the USDI. Why reds? Well, red mangroves also, they grow propagules, which has a much larger parental input than the white mangroves, which are seeds. And so I wanted to see if across these species, there would be different impacts of the substrates on the overall growth and survival. And for my main research question, I wanted to see if we could effectively repurpose sargassum and crushed glass to grow red and white mangrove seedlings in a land-based nursery. And my main experimental questions are, what is the effect of the different substrates, potting soil, crushed glass, sargassum, and a mix of crushed glass and sargassum on the survivorship and growth of red and white mangrove seedlings? And then secondly, what is the effect of these substrates, especially adding sargassum to crushed glass, have on the above and below ground biomass of these seedlings when you compare it to the control of the potting soil? And then within these questions, I had some questions. So we had, for the first question, focused on things like survivorship, as well as the different growth metrics like relative growth rates, number of branches, leaves, and nodes. And my second question focused on the above and below ground biomass, as well as different biomass ratios, which is fine to coarse, and then finally roots to shoot ratios. For where I collected these plants, I went out with help around October 23rd and 27th, 2024, across these 5 different sites across St. Thomas. And I collected both mangrove propagules and seeds from Perseverance Bay, Mandal Bay, Besset Bay, Oasis Cove, and Compass Point. And then these propagules were brought back to the UBI nursery, and we removed any plants that had any seeds or propagules that had observable blemishes on them or defects, and those were discarded from the total population of seeds and propagules. And then for the white mangroves, I measured things like the total mass, the seed length, the radical length, total length, and the width. And then for the red mangroves, I measured their total mass, their hypocaudal length, their diameter with this lovely caliper, and then hypocaudal length and total length. Each one was assigned a unit number, you can see right here with the tag, and this was used to track them over a 6-month period in a land-based nursery. So when they were planted in 3-inch tree pots with a shade cloth, and this is because, as you can see in the room, I have some glass, and the glass would very much easily fall to the bottom of the pot. And so this substrate helps reduce this shade cloth, reduce that substrate loss. And each pot was filled to about 17 centimeters because then I would be collecting another metric of decomposition or compaction. And then we have lovely help for sargassum collection. I had Ali help me with that a lot. We got tons of sargassum from intertidal zones, and the aim was to get nicely freshly beached sargassum or worms right in that water column. For setting up the tanks for the experiments, I had my 5 treatments across 20 tanks. And these tanks were broken up by 15 seedlings being placed in each tray. And that's based on the species. So we see that red mangroves are in one tray by themselves, and then the white ones in that tray by themselves as well. And then I had across my 5 substrates being soil, crushed glass, 25% sargassum plus 75% glass, all further known, I'll be calling this the 25% sargassum, then the 75% sargassum and 25% glass, 75% sargassum, and then sargassum. And you can see here an image of how this first mixture of the 25% sargassum looks, as well as the 75% sargassum. And these species, each 2 trays were kept together and rotated across these different tanks bi-weekly to reduce the effect of location on overall growth and survival. For my monitoring, the plants were monitored once a month for survival, whether it's alive or dead. Then I had the different growth metrics such as height, leaves, nodes, branches. And importantly to note, I did nodes because nodes tell you the potential for growth. So the more nodes you have, the more likely it is to continue growing. And the more leaves it also get. And then for environmental variables, I measured the dissolved oxygen once at the end of the experiments. Temperature, salinity, compaction, and predation was also measured once a month. For biomass ratios, at the end of the experiments, I sacrificed about 86 plants, and I topped them up and put them in a dryer, an oven, for about 20 days, and I was able to get the leaf, stem, and different root fractions like fine and coarse roots. So we did this monitoring, and what we found was that there were baseline differences. So the collection sites impacted how these plants grew, well, how they differed at intake. So for the white mangrove or L. racemosa on the left, this is a radar plot that's showing you the different sites. So we have Mandal Bay, Perseverance Bay, Bessa Bay, and it's showing here that the Mandal Bay plants had smallest measurements across these intake metrics when compared to sites like Perseverance Bay. And then for the red mangroves, we have here, this is Compass Point, Oasis Cove, and Perseverance Bay. Compass Point had the smallest mangroves, and while Perseverance and Oasis Cove were much larger across these metrics. And so because of these differences, I then had to include site in the downstream analyses wherever it was applicable. Reminder about a hypothesis for survivorship. So I expected to have this ranking of soil being better than glass, being better than 25% sargassum, 75% sargassum, and 100% sargassum. But is that what we found? Well, for survivorship, we found that there was a significant difference in survivorship for the white mangroves here on the left, with soil doing the best when compared to the other substrates. While for the red mangroves, the soil, glass, and 25% sargassum did pretty good overall, and they were significantly different to the 75% sargassum. So what this means is that the red mangroves are much more resilient in terms of the different substrates. We can see here an example of a very resilient red mangrove. Early on in the experiments, it was chewed at the stem, and it was still thriving near the end of the experiments. And then to see how this growth happened over time, for the white mangroves, we can see that there was a large amount, substantial mortality at the first month of the experiments, and then continued to decline across all substrates. While for the red mangroves, they stayed fairly the same across up to about 3 months until the 75% and 100% sargassum began to decline. A lot of this early mortality for the caterpillars, for the white mangroves, was caused by these caterpillars. So if you look right here, you can see this caterpillar that had just had a feast on many of my white mangroves. So that's also an implication in terms of survivorship. So that definitely affected it, and not just the treatment. And then another thing that could have affected their survivorship was this compaction of the substrate, as well as the decomposition, especially found in the sargassum mixtures. So this is a line graph showing you mean compaction on the y-axis, with the x-axis showing you the treatments. And the color schemes are the same across all of the plots, where we have the soil being that reddish color, glass being green, the 25 being a green teal, 75 blue, and then 100% sargassum pink. And we see that definitely the decomposition could have affected survival, because shown here in this image, we see that the plants would have started off about this height, and then over time, they got to about this low when they grew. And so that difference in the substrate height could definitely have affected their roots, as well as how they grew with their leaves and stems. Another thing that I tested was the predation probability for the different species. For the white mangroves, the soil and the 25% sargassum were significantly more likely to be predated upon when compared to the glass and 75% sargassum. So our biggest pest was definitely with caterpillars. You can see him right here having fun on this leaf. While for the red mangrove, there was no significant difference of predation probability across all these different treatments. Overall, for survivorship, we found that there was no significant site difference across both species, and I expected to have this ranking, but it was much more nuanced, and so I was able to then reject the null hypothesis. And what this means is that you can use fresh sargassum or fresh glass to grow red mangroves, but that reds are more resilient when it compares to the white mangroves. And then this is where we see that they grew. We want to see now, how these growth metrics added up. So this is me. I take a bird's eye view when it came to these monitoring measurements. Et al. 2022 paper, they found that there was highest survivorship in the 75% sargassum compost when compared to the other different compost mixtures. And so then I expected to have a similar outcome. But let's see. We found for growth in terms of height, overall, we found definitely that for the white mangroves, racemosa, that the height was greatest in the soil and lowest in the glass. And then the sargassum mixtures were pretty high as well in comparison. Well, for the red mangroves, it was soil doing best, glass and 25% doing about the same, and then the other two mixtures being significantly lower. Both species, we found that soil did best. And another metric that we found, one more thing to add to this is that you can see here that for the amount of growth, a lot of it started to occur after 3 months, but then it started to peter off, which could potentially mean that there is limited nutrients left in these other substrates, especially right here. That was branches, and for branches, we found that for white mangroves, the soil did better than the glass. So we see here, soil did better than glass, and 25% sargassum, but not so significantly different from the 75% sargassum. And then 100% only had one guy survive, so did not do stats on one guy. And then for the red mangroves, soil did better than everybody else. And then there was much lower numbers of branches in the compass point seedlings. And we saw that before, that at the beginning compass point, red mangroves were smaller than the other sites. And then for our leaves, another metric of how much they grew, we see here that the white mangroves grew greatest in soil versus the glass. And then there was no significant site difference. And then for the soil and the red mangroves, we see that the soil did better than everybody else. They got the most leaves grown. Another metric I did was I mentioned the nodes. And so how I did the node measurements, which is different to how we generally do it, is I measured at the joint where the hypocaudal and epicaudal meet because of things like this, where it got decapitated, and then I would have had no node to count. So that accounts for how resilient mangroves are in terms of dying. And then for my node measurements, I found that the soil did better than the glass and 25% sargassum for the white mangroves with weak site differences, so the potential for growth could be affected by certain sites across both species. And then for perseverance, soil and other treatments did no different in terms of survivorship, with perseverance providing plants that had the most nodes. So we figured out, that they grew faster, right? But want to know how much faster they grew. And so, or how different these plants were able to allocate these nutrients. And we're going to look here at the above and below ground biomass for in the interest of time. So for the different biomass metrics, I randomly selected 2 to 3 plants from each replicate and treatment. Like I said, I aimed for 100 plants, but mangroves don't usually survive, or they don't follow the order of things that you set for them. And so I ended up with 86 plants. And another limitation that I found in my drying process was that there was moisture absorption due to the large paper bags that I had to then remove even more plants from the overall number. And that led me to have. That led me to have 65 plants remaining. And so here we can see the overall above, so this is a plot and it's mirrored along the 0, with above being the above ground biomass, which includes the leaf and the white and the stem in the light gray. And then below the plot we have the low ground biomass, which is the coarse roots, and then the fine roots. And so here we can see that I lost a bunch of plants in the glass because of those negative values, negative biomass values, as well as only 100%, so that wasn't even included. So that's where it looks different. And then for the white mangroves, I can see here that the soil ones were significantly different in terms of above ground biomass when compared to the 25% sargassum, but not significantly different across the below ground biomass. So both 5% coarse roots overall, those combined mixtures were not significantly different. While for the red mangrove seedlings, we found that the soil was significantly different across both above and below ground biomass when compared to all the different other metrics. Different treatments, sorry. So this is showing that here. And then I want to see how that below ground biomass is broken up into the fine and coarse roots to see if these differences are significantly different. And so what we found was that there was no significant difference across the below ground biomass for the white mangroves, which suggests that they were able to equally distribute their fine and coarse roots. While for the red mangroves, in the soil fine root system, it was more significantly greater than the glass and other treatments. And so this is just also, it's also supported by the Farnaberger et al. Paper that I showed at the beginning that showed that the fine roots were significantly affected in the glass measurements when compared to the soil. And it wasn't supported for the white mangrove, but that's where there is a species-specific difference. So with that, I was able to then reject the null hypothesis that there was no significant difference with substrates across the above and below ground biomass. And then that's because the above ground biomass agrees with sargassum, as well as the fine roots being most affected. So we figured out that they grew differently, but how fast did they grow differently? And to test that, I did the relative growth rate. Relative growth rate is showing how they grew over time with a count of their initial sizes, while absolute growth rate, or AGR, is showing you just total growth, so how much they grew over time. And then here we also have similar thing with our soil being that green, 25% being that yellow, 75% being the glass, orange, and then glass being blue, 100% being pink. We can see here that for the white mangroves, that there was a significant difference in the soil absolute growth rate versus the 75% sargassum mixtures, while for the relative growth rate, the 75% sargassum was significantly greater to the soil. However, keep in mind there were only 3 remaining plants, so that may be different if we had more plants to compare across. And then for the red mangroves on the bottom, absolute growth rate, where it's much more mixed, we see that the red mangroves in soil were significantly different across everything else, so significantly greater, except for the glass. While the different sargassum mixtures found that there was no significant difference. While for the relative growth rate, soil did better than the 75 and 100% sargassum, but not significantly different. Well, about that. Greater than the glass and 25, but not significantly different from the 75 and 100. And generally, soil did best across soil produced the fastest growing seedlings. Summarize all this, it's a lot. Where I expected a certain ranking, I did not get the same ranking across the different metrics. So across relative growth rate, branches, leaves, nor nodes, it was much more nuanced. And so with that nuance, I was able to then reject the null hypothesis, and because that soil performed generally the best. And then we had potentially different things that could have caused it. So what could have caused these differences that I found? Well, to test that, I measured some environmental factors, and I'll be focusing on the dissolved oxygen as well as the salinity. So for salinity, the tanks actually became hypersaline at the end. So this is a line graph showing the salinity in parts per thousand on the y-axis and the date that the monitorings were taken. And then we can see here that we started at about 35, which is generally seawater level, and that increased to above 40. And that became hypersaline. And what we found was that research from Kimura et al. 2024 found that plants did best in about 50% seawater. So my salinity got much higher, and that could have been due to evaporation caused by them being open flow tube system, right? And then they found that the optimal ranges for well-developed mangrove stands in Florida, the salinity would range from 24.5 parts per thousand to 33.5 parts per thousand. So I would suggest that we mix it with seawater and fresh water to get that ideal number. And then another thing is that for our dissolved oxygen, we have this red line that's showing the level where they become anoxic. And we see that not many of them fell below that line except for the white mangrove, which is the green, while the red is the red. And so with a low dissolved oxygen within these pots, it could affect the physiological and morphological growth patterns of these mangroves. So basically how the mangroves look and then how they perform. So because these numbers were generally pretty low in terms of dissolved oxygen, that could affect their growth and survivorship. And we found that to these different salinity and dissolved oxygen metrics, that the white mangroves are much more susceptible to these differences.